Biology. Hello everyone, welcome to Assist Biology. In this video, we will discuss about post-fertilization events, and this is the fifth part from this chapter, sexual reproduction in flowering plant. If you have not seen the first four part, I will mention the link in the description box so that you can watch the video. I am Ashish Kumar Das, a student of MSc Animal Biology, University of Hyderabad. Now let's get started. So first of all, what we will discuss today? Okay, so we will discuss what are post-fertilization events, what is endosperm, what are the different kind of endosperm, that is the function of endosperm. Again we will discuss the function of endosperm. We will see what is embryo, how is it formed, then we will see monocot and dicot embryo and many more. So post-fertilization, as the name suggests, post-fertilization means these events occur after fertilization or occurred after fertilization. So all events after the double fertilization are called post-fertilization events. The example are development of endosperm, development of embryo, that is the maturation of ovule into seed, and the maturation of ovary into fruit. Now endosperm development. So what is endosperm? We have discussed that is the triploid nutritive tissue. So endosperm development precedes the seed development. So endosperm is formed before the embryo development begins, or the endosperm development begins before the embryo development. And the three modes of development are nuclear, cellular and holobial. Okay, we will see all of these: nuclear, cellular and holobial. Most common is the nuclear type, that is the triploid endosperm nucleus undergoes repeated division without cytokinesis, and this results in free nuclear endosperm. So in the nuclear endosperm you can see there are a number of nuclei formed, and these gradually the cytokinesis occur after the karyokinesis, and they gradually form the cellular endosperm. Here the each nuclear division is not immediately followed by cellular division in the nuclear endosperm, but in cellular endosperm each nuclear division is immediately followed by cellular division. And in holobial endosperm, holobial mode of development, the both cell are divided by division, and in one the nuclear endosperm is taking place or cellular endosperm is taking place, and in another the other mode. So here above the cellular endosperm mode development, and here the pre-nuclear mode of development. You can see here the nuclear mode of development: one nucleus, two nucleus after mitosis, number of nucleus, and always after the number of resulting number of the nucleus, the cellular formation or the cell membrane formation always start from the periphery. That's why in the center free space is there. You can see this is the pre-nuclear mode of division. So endosperm development, cellular formation from the periphery, completely cellular, it will become completely cellular after some repeated cytokinesis. Example are coconut, rice, maize, sunflower. And what is the function of endosperm? Function of endosperm is to provide food for the developing embryo. And on the basis of the embryo remnant or embryo remaining, the endosperm remaining, we can classify them into non-albuminous, that is without the endosperm, and the endosperm is completely consumed during the development of the embryo. Example are pea, bean and mustard. Example are always important. And in the albuminous seed, a portion of endosperm remains. So example are castor, maize, coconut and rice. Okay, you have to remember all the example and the name, how they means, what does they mean, that is the non-albuminous and albuminous. We have covered the endosperm. Now come to embryo. So embryo development start after certain amount of nutrient is formed, that is after the endosperm formation begins, and this is to ensure the nutrition supply to the developing embryo. And this, first of all, what happens, you see the chronology. In the zygote by mitosis the proembryo is formed, proembryo divides to form the globular, globular divide to form the heart shape, and this heart shape divides to form, that is undergo development to form the horseshoe shaped embryo, that is the mature embryo. You can see the diagram here. This is the embryo sac after the division, the zygote, that is 2n in nature. Then the zygote is forming proembryo, proembryo is forming globular embryo, globular is forming the heart shaped embryo, and the heart shape is forming the horseshoe shaped embryo. I have taken this diagram from NCERT textbook class 12th. So you can see here the enlarged embryo sac after fertilization, primary endosperm nucleus 3n is there, and the zygote 2n is also there. You can see it. Dicot embryo, here what happens, two cotyledon, as the name suggests, dicot means two cotyledon. Here the two cotyledon are there, and what happens, the portion of embryonal axis above the level of cotyledon, you can see these are the two cotyledons. Above means this portion, okay, The above the level of cotyledon is called epicotyle and it terminates in the plumule. Okay, here this is the plumule. So this is the shoot apical meristem or shoot meristem. That is, they will form the shoot after the seed germination. And the portion of embryonal axis below the level of cotyledon. So this is below the level of cotyledon, and they will form the radicle. Okay, and the below the level of cotyledon, that is called as hypocotyle, and it terminates in radicle. Radicle is the root tip, that is, it will lead to root after the seed germination. So above is plumule, below is radicle. Now come to monocot embryo. The previous one is dicot embryo. Now come to monocot embryo. Monocot embryo occurs in grasses and it is having only one cotyledon, that is called scutellum. You have to remember scutellum, and it is pushed towards one side of the embryonal axis. Radicle is on the lower end, as you have seen. Radicle is on the lower end, and the plumule inside the coleoptile is the plumule. Plumule is at the upper end. The plumule, that is at the upper end, or the epicotyle you can say, it is covered by a hollow foliar sheath called as coleoptile. You have to remember this. Plumule is covered by coleoptile. Okay, and radicle is covered by coleorhiza. You have to remember this. Now you can see here the structure. One is the dicot seed, that is your common bean. Here the seed coat you can see, plumule, that is forming the shoot, radicle will form the root, and here these are the two cotyledons, epicotyle, hypocotyle. Epicotyle means above the level of cotyledon, hypocotyle means below the level of cotyledon. You can see here, this is the dicot seed. And a monocot seed, that is corn, you can see endosperm is there, radicle is there, radicle is downwards, plumule is upward. Okay, plumule is, coleoptile is covering the plumule, and coleorhiza is covering the radicle, and scutellum is the single cotyledon of the monocots. So here you can analyze the difference between a dicot embryo or dicot seed and a monocot seed, and you can see here the orientation of the cotyledons, epicotyle, hypocotyle, etc. Now seed. So what is seed? Seed is a mature or fertilized embryo. The most precise scientific definition of seed is it is a fertilized or mature ovule, and it consists of seed coat, one or two cotyledon depending upon whether it is monocot or dicot, and an embryonal axis. So seed coat is a double layered structure, and it is composed of testa and tegmen. Testa is formed from the outer integument and tegmen is formed from the inner integument. You have to remember this. All the structures that are in the ovule or the before fertilization will convert into some structure in after fertilization. That is, the outer integument will form the testa, inner integument will form the tegmen, and a micropyle is a small opening to ensure the entry of water and oxygen. Hilum remains a scare on the seed, we have already seen. Cotyledons that are thick and with reserve food. So what are the function of cotyledons? After the when the endosperm is not there, cotyledons will act as the source of food. In most of the seed, endosperm is completely consumed during embryo development. So now the cotyledon will come into effect and they will act as source of food. And albuminous and non-albuminous seed we have already discussed. That is, albuminous means with endosperm, non-albuminous means without endosperm. Thus, what is perisperm? Perisperm is the remnant of nucellus. Sorry, this will be nucellus. Okay, so this will be nucellus. So remnant of nucellus is the perisperm. Okay, and this is found in black pepper, water lily, beet, etc. So perisperm is found in where? Water lily, black pepper, and beet. And what is perisperm? This is the persistent nucellus or the remnant of nucellus remaining still the or after the seed formation. As the seed matures, what happens? The water content decreases and the seed becomes more and more dry, and there is a state of inactivity that is called as dormancy. Seed dormancy, you must have heard. This is usually, this can be overcome by rubbing the seed with a sandpaper or by cutting the seed with a knife, etc. So seed dormancy is the state of inactivity. Now what are the advantages of seed? These are most important part. Advantages of seed, we will see what are the advantages to plant, and we will also see what are the advantages to human or mankind or our civilization. So first of all, seed having a better adaptive strategy for dispersal to new environment. So as seed are very small structure, they are covered by seed coat, they can be better dispersed with less chances of being lost. Okay, so why they are important? They are better adaptive strategy. Then reserve food, they are having reserve food in the form of endosperm to nourish the seedlings, that is after germination. Then seed coat will provide protection, this will be protection to embryo. Okay, seed coat will provide, this will be protection, this will be protection, not nourishment. So seed coat will provide protection to embryo, and as they are formed as a result of sexual reproduction, that is the fusion between two gametes, they are having chances of genetic variation that will lead to evolution. Okay, that will always help in the continuity of species or development of more better species as a result of evolution. Then they do not depend on water for fertilization. Okay, as they do not depend on water for fertilization, so the formation is certain because no special condition are needed for seed formation. Okay, and this will ensure the continuity of species. As you have seen in bryophyte, water is required, so if there is no water, they can't undergo fertilization, they will not fuse. That's why here the problem is overcome by forming seed. So what are the advantages to mankind? They stored and used as a food throughout the year. Okay, so as they are small structures, they can be easily stored in grains, as grains, okay, in containers or in carry bags. Okay, then used to raise crop in favorable condition, as the seeds have ability to form new plant under germination. So structure of maize grain, in many exam question come from this, so I thought to put this. So what happens in grass family, that is in maize family, maize, rice, wheat, etc., there is a single seeded fruit, and this fruit is called as grain. Okay, and the pericarp and seed coat form the husks. You can see the husks in rice. From paddy we remove the husks to form the rice. Okay, so here you can see the pericarp, the aileron layer is there, it stores the protein. Okay, the endosperm is there, the scutellum is the single cotyledon, coleoptile plumule, that is the plumule is the pseudapical meristem. Inside the plumule, pseudapical meristem is there, it is covered by coleoptile, and inside the coleorrhea, that is radicle, and radicle is having root apical meristem. Aileron layer store protein, this is important. Now fruit. So what is fruit? This is the matured ovule. Okay, after the fertilization, ovule transformed to, matured ovary. Ovule matured to seed and the ovary matured to fruit. Ovary enlarges, the size increases, ovary wall become the fruit wall, and this is called as pericarp. The fleshy pericarp is found in guava, tomato, cucumber. You have seen the wall of the fruit is here, the fleshy, that is in guava, the fruit we consume, in tomato, in cucumber, and leathery and dry pericarp in pea, in bean, in mustard. You must have seen these fruits in your life. Pericarp protect the seeds and help in dispersal. So pericarp is the fruit wall you have seen. So this is above the seed, or this is above the above the pulp of the fruit, you can say. So this always help in the protection of seed and help in their dispersal. And what are parthenocarpic fruit? The parthenocarpic fruit are the fruits that are developed without fertilization and they are seedless. Okay, example are banana. And how parthenocarpy can be induced? It can be induced by parthenocarpy can be induced by auxins and gibberellins. Okay, so parthenocarpic fruit formation can be induced by auxin and gibberellin. These are the phytohormones. So what are false fruit? Again, another important concept. False fruit is a part other than the ovary. Here what happens, if any part other than the ovary contribute to fruit formation, then it will become false fruit. Example are apple and pear. We will see the diagram. Parthenocarpic fruit example banana, and false fruit example apple and pear. You can see the true fruit mango, the epicarp, mesocarp, endocarp is there, seed coat and seed is there. So this is a true fruit. Here part other than ovary do not contribute to the fruit formation, but here what happens, the false fruit, the part other than ovary contributes. So pericarp is there, seed is there, fleshy thalamus is there. So fleshy thalamus is coming from other than ovary, that's why this is a false fruit. The thalamus contribute to the fruit formation in the apple, and you can see these are the remnants of calyx. Okay, so false fruit example is apple, and here the thalamus contribute to the fruit formation. This is the end of the video. Thank you for watching this.